The title of today's talk is Design and Evaluation of In Situ Bioremediation Applications. And again, my name is Brad Elkins with EOS Remediation, and I will just go ahead and get started. For those of you in the audience who maybe don't know or would like a, kind of a refresher, what is bioremediation? So we're utilizing naturally occurring organisms in the subsurface in an aquifer to clean up contaminants of concern. And in most cases, you have to stimulate those microorganisms. You have to give them either a food source or you have to give them something to breathe. And you need an electron donor or an electron acceptor. And in a lot of cases, the electron donor can be just naturally occurring organic matter in the subsurface. But more than often, it's something that has to be added to the system, like a sodium lactate, molasses, or emulsified vegetable oil. And then on the electron acceptor side, again, that's something that you might think of as a respiring agent, so oxygen. And then, of course, if it's an anaerobic organism, it can utilize nitrate, iron, sulfate, and chlorinated solvents. And there's really two things that occur. So when we're talking about remediating chlorinated solvents, that electron donor that you inject into the ground is fermented. So a little bit of a misconception that I have heard is most people refer to EOS as the electron donor, and that's not true. EOS is the source of electron donor. The electron donor is actually released during fermentation of the oil, and this is true of any carbon source. So if we were talking about lactate, for example, the lactate is not the electron donor. It is the source of electron donor. It is fermented into hydrogen and acetate. And then there's respiration of that occurs within the microorganism itself. And just this little animation kind of brings it all together. So you inject your EOS, it ferments into molecular hydrogen and volatile fatty acids. D-halococoides, along with several other D-halo-respiring organisms, can utilize that electron donor, and through microbial respiration, exchange hydrogen with a chlorine, and in the case of PCE, degrades it to TCE. And then this would continue until eventually you get to ethene. And most of you are very aware, and I promise I will keep the sales pitch to a minimum here. But basically, this is kind of where we shine. We're sort of the experts in making this reaction occur at your site. We supply long-term electron donor sources, that's EOS. We provide the BAC9 culture, which is the D-halococoides, along with a bunch of other of his friends, and then the CoBuff MG product, which is a magnesium hydroxide based amendment to either adjust your pH or maintain pH at a neutral environment, because that's important for this process to occur. Now, those of you in the audience who maybe don't do a lot of chlorinated solvent type work, maybe you do more petroleum work, well, we can help you there too. The difference is that now the role slightly reverses. The electron donor is usually now very abundant, and with time, especially at gasoline release sites, you typically have very reducing conditions because there's a lot of carbon there that can be utilized. So the organisms there have already burned through whatever available electron acceptors there may be. And so you have to add something like oxygen or sulfate to the system to stimulate those organisms to keep them going. And we do that with either EOX or EAS. When you're thinking about a project and you're trying to choose the right type of carbon substrate, and most of this next part of the talk is going to focus on selecting electron donor sources for chlorinated solvents or hexavalent chromium or perchlorate or something of that nature. But some of this is easily transferable to electron acceptors or to other carbon sources that may be out there. The first thing to consider, take into mind, is how does your site sort of lay out? Are you concerned just about the source area? Do you want to just try to knock down some really high concentration source? Do you have a biobarrier? Maybe you have a plume that's starting to migrate off site, or maybe it's already done that, and you want to sort of control that. You want to either stop it from happening or maybe cut the plume in half, dissect it. for whatever reason. And so maybe you want to attack the plume in sections with biobarriers, or maybe you want to do both. Maybe you want to do source area treatment and do some sort of plume control through some type of barrier. So that's kind of step number one. How does your site laid out? And then obviously the next couple of things they're not really groundbreaking, but they are things you need to take into account. Your geology is, going to be very important. I mean, how your strata is laid, your silts, your clays, your gravels, these are going to determine your distribution of the product. And they're also going to influence heavily on your injection rates and what's really going to be possible in terms of distribution of the electron donor source as you inject it into the subsurface. Probably one of the most common questions that I get is, what's my injection rate going to be of this stuff? And you really can't answer that just by looking at boring logs or looking at some site data. That's where a pilot study is really going to be beneficial, is to test those kind of things. And in a lot of cases, some people may not necessarily inject amendment in their pilot study. They may just go out and do, a water injection, some type of water test to really determine, what the storage capacity of their aquifer is and how much they're really going to be able to get into the ground. Groundwater velocity kind of goes hand in hand with the lithology a little bit, in that if your water is moving really fast and you want to do a biobarrier, how you engineer that biobarrier is critical. It will dictate the longevity of any product where you inject it. More velocity means more mass flux through your treatment zone. So understanding that is very important to not only selecting the right substrate, but dosing it correctly. These two kind of go in hand, oxidation reduction potential and then your competing electron acceptors. So if you recall the illustration I showed at the beginning, I showed oxygen, nitrate, sulfate, iron, and then the chlorinated solvent. So there is a preferential order at which these electron acceptors are consumed in the subsurface. And again, this is not really news to anybody, but it is, again, it's something that kind of comes up when we're talking about how much electron donor source you will need. Understanding where your site is in that redox ladder and how much competing electron acceptors you have, it is critical for properly dosing your site. I have heard people say that, well, my site's very aerobic, and I really don't think in situ bio via anaerobic enhanced reductive dechlorination would work. And that's not true. EOS has a lot of reducing equivalents in it. It can take an aquifer that's aerobic and turn it anaerobic very easily. So I wouldn't rule out a site just because it's got a very positive ORP or maybe a lot of DO or even a lot of nitrate. Just we're going to need to take those things into account when we dose the site. When we do our engineering calculations, those are things we're going to want to know. But it is certainly not something that is a limiting factor. Commingled contaminants. This is something else that seems to come up a lot, is you've got maybe a petroleum hydrocarbon intermixed with your chlorinated solvents. Maybe something like benzene intermixed with carbon tetrachloride, or, there's tons of combinations out there. What's important here is that recognize from the beginning slides that EOS, or a carbon source, is serving the role of an electron donor, and your petroleum hydrocarbons are an electron donor too. So you can't add EOS to treat both types of contaminants. And if you add EOS and you also eventually want to take care of that benzene, you need to understand how you're going to dose that site, where it's going to get dosed, and recognize that eventually you're going to have to flip that aquifer to maybe, say, sulfate reduction. You're going to have to start adding sulfate. You wouldn't want to add them necessarily at the same time, but coordinating your treatment train, again, will result in success at the site. Your microbiology at the site, this will determine, do you need to bioaugment? Something else to take into account is if you're out there doing your census, trying to understand what your D. halicoides population is like. In some cases, you may not need to inject something like, say, EOS Pro, that's got a lot of nutrients and vitamins going into it, because you've already got a very robust population, and maybe you just need to add a little bit extra carbon to kind of get things going in the right direction, or maybe enhance what's already going on. In a lot of cases, Frank Loeffler pointed this out last week in the Microbial Insights webinar, D-halicocoides really needs vitamin B12 to grow and do its processes, and so there is that to take into account. But if you're kind of at that point in your project where you just need a little bit more carbon to get it to go, is it critical? Those are things you're going to want to take into account. Recognize that different substrates degrade at different times. EOS is not just soybean oil. It's got soluble substrate mixed into it that's going to ferment first to kind of help things get started. Same thing with if you're looking at EVO versus molasses or EVO versus lactate or any, maybe some type of, waste product like a soda pop syrup or something like that. Recognize that these things degrade at different times, and that as long as your project can handle multiple injection events within a short period of time, okay, great. But if you only want to go out there once every couple of years, or again, you've got high groundwater velocity, these are things that are going to impact the overall project cost. Finally, your injection method, and this can be controlled based on site activity. If you're working at a site that is in operation, you may be limited to how you can deliver the amendment. The good thing about EOS and a lot of our other products is that there really is no limitation in terms of delivery. Traditional wells work great because you can get a good, nice seal in the annulus, and that helps prevent daylighting and sort of any type of short circuit up through that annulus. But DPT works very well in a lot of situations. Also, you can utilize existing infrastructure. Say you already had some type of physical treatment in place, like a SVE air sparge system. You could utilize some of that infrastructure to install your EOS. One of the not so common ways of doing it, but is possible, is in placement prior to backfill. That would be more common for your petroleum sites, but it could be done with EOS. This is really, what are you paying for? You want, most of the time, you want a carbon source that's going to produce a lot of electron donor per pound or per unit mass. Here I'm showing it in the red graph on the right-hand side as per kilogram. So I'm expressing it as a molecular hydrogen per kilogram of substrate. And you can see that of the various common carbon sources, electron donor sources that are put into the ground, soybean oil just by far, outperforms all of them, which is one of the reasons why we use it so much. On the flip side, if you're looking at how the substrate will behave beyond just an electron donor production, you also got to consider acidity. This material will ferment, it will produce acid, and you will also produce acid by dechlorination. So trying to reduce the amount of acidity in a short period of time would be ideal in keeping the aquifer at a relatively neutral pH. So the graph on the left is showing the acidity. We're expressing it as the H ion per molecular hydrogen that's produced. And you can see that soybean oil, again, doesn't produce a lot of acidity over its fermentation life, especially when you compare it to something like lactic acid or ethyl lactate. Keeping this in mind during your project is important. If your alkalinity is really low at your site, this is definitely going to be something that's going to impact your performance long term. Why is this? Why does soybean oil have such high hydrogen and such low acidity? You would think that the more hydrogen it has, the more acidity it would produce. And that is true to a point. The difference is really how that acidity is released. So if we look first at soluble substrates, those are going to go through a very quick fermentation process, say over the course of a few weeks or maybe a couple of months. And they're going to quickly ferment into acetic acid or acetate and hydrogen. And that is going to create rapid decrease in ORP and create reducing conditions very fast. Because it occurs so quickly and in such a short period of time, you can get this sort of initial shock to the aquifer, and you can see an abrupt drop in pH. When we compare this to slow release substrates, These ferment over sort of a multi-step process. The soybean oil will first hydrolyze, it will break down into glycerol and long-chain fatty acids, and those will break down further into various volatile fatty acids, which will break down into molecular hydrogen acetate. And there will be some acidity produced during that reaction as well. The big difference is that because it kind of goes through this multi-step process of fermentation, this occurs over a very long period of time, years. Because of that, the acidity is released very slowly, and there isn't this sort of initial shock or anything like that in pH. That isn't to say that you won't see any decrease in pH if you used soybean oil, but that pH will occur somewhat slowly over time, and you can have an opportunity to deal with it before it really becomes a major issue. So you bought some EVO, you got some EOS, and how do you inject it? And how are you going to deliver this amendment into the subsurface? Obviously, I talked about various well construction methods, DPT versus temporary or permanent wells. But beyond that, you can set your even with the source area treatment, you can set those points up in a grid type formation or even barriers that have some space in between. I would say that there's two main ways that I typically see EOS being injected. The first, if you look at the pictures on the right-hand side up in the top corner there, you'll see a dosatron. So that's a non-electrical, hydraulically driven chemical feed pump. So the hose there you see connecting into the pump there, that's that green hose going into the side at the bottom. That is a source of water. So you would hook up your source of water into this pump, and that action of the water flow would drive the pump. It would pull the EOS up from the drum or the tote, and it would mix it in line to whatever desired ratio you had the dosatron set to. These are attractive for a number of reasons. They don't require any electricity. You don't need tanks or anything like that. You just need some hosing and a water source. The drawback is that they really can only be utilized one at a time. They're slow. You can't get really high injection rates off of them. So you're going to be injecting, kind of one well at a time if you use something like this. And that's why that picture of the totes there at the top has, if you can tell, it has several dosatrons on each one, because they're trying to inject into, half a dozen wells at one time. Because if they did it one at a time, they'd be out in the field forever. So that's a drawback. The other way that you can inject EOS is at the bottom. So you see you've got a big frac tank there, a Baker tank or a rain for rent tank. And then you've got a conical mix tank. So this particular client, they had their EOS delivered in a tanker truck. They offloaded that tanker into one of these frac tanks, and then they used a conical tank to mix up batches of their dilute EOS mix, and then they used a centrifugal pump to deliver that dilute amendment out to their various injection wells. And you can see in the picture right above that, they've got it manifolded to several wells. So they're injecting sort of simultaneously 3 or 4 points at a time off of one batch. And of course, with like any injection, whether you were doing it with the dosatron or with a mixed tank, you're going to want some way to monitor your flow at each point, and a way to shut off flow at each point, and also a way to gauge your pressure. Typically you want to do this at a relatively low pressure, something under 10 psi. Sometimes you can get up between 10 and 20 psi if the site's taking it really slow and you want to put some more oomph behind it. But you want to do this in kind of a low pressure, at moderate flow levels. You don't want to slam this stuff into the ground, because that's how you get short circuiting. That's how you create preferential pathways. The EOS is diluted in a variety of ratios. 5:1 to 20:1 is probably the most common that's used. And then once you've injected your EOS, we always recommend, unless you're doing DPT or something, that you follow up with some chase water. I'd still might recommend that even if you were doing DPT, just to be sure that you're getting distribution across the radius of influence that you want. And usually something between 25 to 50% of the effective pore volume within that radius of influence is what you need in terms of chase water. You can go on the lower end if you're concerned about time in the field, but just recognize that as you reduce your volume, you theoretically will also reduce your distribution coverage, unless you've made up that volume in diluted EOS. The other thing about EOS is you can add basically anything you want to it. We provide, like EOS Pro as an example, it's provided as a 60% emulsion of soybean oil, but we also include, phosphate nutrients, vitamin B12 supplement. You guys have probably all seen that in the little 16-ounce bottles that we ship with every drum, or 5 per tote. But you can also add things like buffers, whatever buffer of your choosing. Sodium bicarbonate. There are some things that we can actually add in the factory if you were interested in that. If you wanted, say, a certain percentage of sodium bicarbonate, or maybe you wanted some source of soluble iron, we could add all of that in our factory and then ship it to you so you don't have to mix it in the field. Some things are not going to mix so well with the emulsion, so we kind of tend to do that on a case-by-case basis. So if that's something you're interested in, just ask me and we can help you out with that. In terms of injection reinjection frequency, I'm sorry, not reinjection frequency. I will talk about that though later. Monitoring frequency. This is another very common question. Remember that the product will ferment slowly over the course of months, years. So performance is not going to happen right away. This isn't ISCO. This isn't kind of a, get it in the ground and then all of a sudden your stuff's magically gone. So doing your post-injection monitoring on a quarterly basis is usually fine. If you want more resolution in the data, you could do something every 2 to 3 months if your budget allows that. But you really don't need to be going out there every couple of weeks or every 30 days. That's not really necessary. It doesn't hurt to always have more data, but if you skipped a monitoring event, or you started doing it quarterly and you were doing it every 30 days, I'd be willing to bet that you're probably not going to miss too much in the data. And I think the most important thing with all of this is that, you always hear that this type of technology is a contact sport. That's usually the analogy that's used, and to a certain point that's true. You want to get good contact between the amendment and where you have the contamination, and that's going to accelerate cleanup. But unlike some other technologies out there, EOS kind of has a fudge factor to it in terms of distribution. As long as you're getting it in the general area of where you need it to go, that's usually going to be pretty good. It does not necessarily have to physically touch the contaminant. That's not required. It just has to be in the vicinity. That oil droplet, the microbe, and the contaminant all have to be within the relatively the same space, and usually things will take place. So, to keep that in mind as you're thinking about your injections, as you're planning how much water and how much time in the field you're going to need. Better contact is always going to mean better treatment. So, as long as you keep that in mind, your project should go without any problems. Okay, so how do you calculate how much EOS you need? I will do this for you. I've probably done it for a lot of you listening in bunches of times. These next couple slides will kind of walk you through what I'm doing for you on the back end. And I typically try to explain this to everybody that I work with. But the first thing that we're going to need to define from the start before we talk about anything else is, well, how big is this treatment area going to be? Surprisingly, more often than not than you would think, I actually get calls about how much EOS do I need, or how much product will I need, and what's left out is how big the area is, or how what the treatment interval is going to be. And that's definitely going to be critical information. So even if you have to kind of guesstimate, you're not really sure, maybe you haven't fully characterized the site yet, you haven't fully delineated, or maybe you're planning on putting in more wells in one area and you just really don't have a good grasp on exactly how big that treatment area is going to be, that's okay. Give me your best guess. That can at least get us down the road in a direction. Because otherwise, I would have no way of even beginning to do a calculation like this. The other thing too is that if you've worked with us, is you've noticed that when we do our design tool, we give you two numbers. We give you an amount of EOS that's based on the hydrogen demand, the amount of oil that's required for biodegradation, including competing electron acceptors. So that would include that high sulfate or nitrate or whatever you have at your site. So that's just a stoichiometric number. You have this amount of mass of contaminant, and you need this amount of mass of oil to degrade that contaminant. It doesn't take any of the other site parameters into account. It is just a mass relationship. Okay. The other number is the oil required based on the oil retention of the aquifer. So based on your groundwater flow, your soil type, your theoretical oil retention capacity of that soil, we will generate an amount of EOS. Now, this more has to do with the physical characteristics of your site. You know, if you have a clay aquifer, you're going to have more surface area. There's smaller pores, and there is more coverage. There's more attachment sites for that oil droplet to stick to. So therefore, you would need more EOS to fill up all of those sites to get that good coverage that you need. What's interesting about that is that, well, at most clay sites, you might need a little bit more EOS, but you can't necessarily get all the time that greater volume into the ground. Well, that's where you're going to have to install your points closer together. You're going to have to reduce that radius of influence. So there's this trade-off between amount of substrate and the number of points, and then also the amount of water. Dr. Bob Borden has given webinars in the past that, he basically says that over the course of like 4 or 5 slides is really what this comes down to is the amount of oil you need based on your oil entrapment, but then how you deliver that substrates based on your injection methodology and the amount of water you're willing to put in the ground. So once we kind of determine that, we can help you figure out how many wells, the spacing between the wells. We can sort of provide some guidance in that area. One thing that can be misleading when you get a design summary from us is because you get 2 numbers of the amount of oil, you don't sum those together. It's the higher of the 2, because the higher number you can assume satisfies the requirement of the other. So it's not a sum. It's just you pick the higher of the 2. And so this is that oil retention calculation. So this is actually pulled directly from the inside of the ESTCP design tool, which is very similar to the one we use. So you're taking your treatment area, your effective porosity, your soil bulk density, and your oil retention. And when you work all that out, basically the feet cancel and you're left with pounds. And so that's how much EOS you would need based on oil entrapment. And again, oil retention is the amount of oil that sorbs or sticks to the aquifer matrix or the aquifer grain. And the idea is you want to fill up as many of those attachment sites as you can, because then that puts more oil in that pore space for treatment. And what controls oil retention? Pretty much the biggest thing is droplet size. Zeta potential also plays a significant role too. Typically clays are going to have a more net negative charge. So if you had a positively charged amendment, that's going to have a greater affinity to stick to that clay. EOS is typically emulsified with non-ionic surfactants, so it's going to have a negative surface charge, which means you're going to typically get a little bit better transport when you have more clay in the aquifer. So understanding the properties of your emulsion and how your emulsion is made can be critical to distribution properties within the aquifer. Don't necessarily be misled just because somebody's selling a cheap EVO. You want to be sure, how is that EVO being prepared, and how is that going to affect its sorption in the aquifer. Probably the 2 most contrasting products that we have right now is this waterless oil product, which is EOS 100, and then EOS Pro, which is like a more traditional EVO. And I've sort of listed out the characteristics here of the 2 products. The biggest thing I think I want you to focus on is the picture at the bottom that shows this is a microscope slide image that I actually took here in my office. This is a diluted EOS 100 at the bottom left and diluted EOS Pro at the bottom right. And you can see the scale there. That's 1 micron droplets in the EOS Pro and about 5 to 10 micron droplets in the EOS 100. So you can see that these products are going to have significantly different retention properties in an aquifer. And to sort of prove this, we send our samples off for particle size distribution all the time, and this is just one of the curves that we generated from a couple of batches. So you can see the EOS 100, again, very heterogeneous particle size distribution. Most of them are falling between the 5 to 10 range. You got a few outliers that are a little bit larger. You look at EOS Pro, pretty much everything is falling below 2 microns or below 1 micron, somewhere within that window. This is the difference between a factory emulsified product and a product that you might make in the field. The little picture on the right hand side is how EOS 100 would be prepared. So this is obviously much different than what we would do in our factory, using very high mixing capabilities to produce a very homogeneous product. This is produced a little more crudely. And it has its advantages and disadvantages depending on your project site, but just want everybody to be aware that there can be a significant difference between those two products, and that you shouldn't necessarily compare the amount of oil apple for apple, because how it will retain in your aquifer is drastically different. We actually did some experiments internally, and we packed a couple of sand columns. We did a silty sand column, which would be representative like a field sand, and then we did a clean sand, which would may or may not exist in nature anywhere, but just to kind of give you an idea that when you have these really clean, coarse-grained sand aquifers under really high flow conditions, it can create substantial differences in how the products behave. And so when we look at the measured oil retention from those two columns, we look at the clean sand, again, that's that masonry sand, versus the field sand, which is the silty sand. You can see that there are dramatic differences in oil retention. Probably the most so being between EOS Pro and between EOS Pro, between the two lithologies there. You're talking a couple of orders of magnitude in difference in oil retention. EOS 100, just because it's so much larger in droplets, yes, it has an increase between the two lithologies, but it's more pronounced than the EOS Pro, simply because with more clay, again, you can get more retention. So how does this affect your project? We did a couple of design models in the ESTCP emulsion toolkit, and this design tool is available free on the ESTCP certif website. Just Google emulsion, ESTCP emulsion toolkit, and you'll get a link right to it. So we looked at a barrier application under a really high groundwater velocity, 1.6 feet per day. So this would be like a medium sand with maybe some silt in it, but the majority of the flow is going through, a medium sand. And initially, when we kind of first did our calculation and we looked at the two products, Pro and 100, next to each other, so we're looking at the graph on the left, we're looking at total project value. We're looking at total cost of the project, so not just substrate. This is everything. This is your fixed costs, your permitting, your substrate, your injection labor, your installation of your wells, any analytical that may need to be done. This is everything to get the product into the ground. And initially, you see that EOS Pro is actually a little bit cheaper than EOS 100, just because you don't need as much substrate, because you're getting really good distribution. You're going to be able to get that product to go across that ROI very effectively. But then when you start looking out 20 years down the road, or over the life cycle of the project, you can set it to whatever you think is realistic for your site. We picked 20 years just because for a lot of sites that's typically fairly realistic, especially for a barrier application. When you start weighing into the fact that groundwater velocity is really high and you have a lot of mass flux running through that barrier, you're going to discover quickly that EOS Pro, even though it got good transport, doesn't have very good retention, is going to wash out and get burned up really fast. So you're going to have to go back out in the field a lot, like every 1.5 to 2 years, and re-inject the Pro, versus the EOS 100, which, yes, you need a lot more product initially, but long term you don't need to go back out as much, maybe 1 or 2 maintenance injections over a 20-year life cycle. So this is something that we always try to educate the consultants on, is be sure that you're not only looking at this as a one-time application, especially if you're doing something like a barrier that's going to be sitting there for a long time and dealing with a lot of mass flux over the next maybe decade or two. How are you adequately engineering that site? Are you adequately selecting the right electron donor source for those hydrogeological conditions? And they can have a significant impact on your bottom line. I don't know if anybody was listening in on that Mythbusters webinar that Microbial Insights did last week, but that was a really good webinar, and they have it recorded on their website. So I would suggest going there and listening to it if you haven't already. But I'll actually quote John Wilson here, because he said that this phenomenon that I'm showing you here, he verbatim said, the goal is to basically mix your amendment with the contaminant water, and the best way to do that is to get your amendment to mix, and he was referring to high velocity aquifers when flow is really a condition. And so by having a product with really good retention is going to enable you to do that. So now you've got your product in the ground and things are humming along nicely. So you want to know what you need to look out for in terms of performance data. Probably some of the most common metrics that are tracked, beyond your field parameters like DO and pH and stuff like that, is going to be, your gases, your fermentation gases, methane, ethene, and ethane, your contaminants of concern, of course, and then another common one is your TOC. So I'd like to talk about TOC for a little bit, because this comes up almost every other day. I think a lot of people, especially when they're getting into bio, or maybe they've had a lot of experience with bio, but maybe it's been more towards injecting soluble substrates. TOC is a very important metric for soluble substrates because it's soluble. So when you take a groundwater sample, you're going to get a very accurate measurement of how much electron donor source is left in that groundwater. And gauging that against your contaminants of concern can indicate when you need to go back out and re-inject. And so typically with soluble substrates, you're trying to maintain this level of TOC in the ground between, say, 50 to 100 milligrams per liter. And then maybe in some cases, given the parameters of your site, maybe you want it higher than that. But either way, that's your performance metric with soluble substrates. People make the mistake of applying this metric to emulsified vegetable oil, and that's not really a good metric. And the reason is because the oil droplets are retained on the aquifer matrix, so they're not in groundwater anymore. You cannot accurately measure TOC from a traditional groundwater sample and then run TOC on it. 99% of the material is sorbed to the aquifer matrix. There is a little bit in that residual in the pore space, so when you sample your wells in the area that's had EOS, you can sometimes see some white milky color in that water, but that is not representative of the mass that's down there. Most of that mass is absorbed to the aquifer matrix. So typically what people see is TOC ranging less than 20 milligrams per liter. Sometimes you can see it around 50 or 100. When you first start your injection, you might see your TOC really high. That's because the oil has not yet adsorbed to the aquifer matrix, but with time you will see it dramatically decrease. And that typically scares people because they think that, oh, well, my EOS or my EVO is gone, and it's not. That's not really representative. The other thing I'd like to just touch on real quick is methane. This seems to be kind of another hot button topic right now in the industry. With EVO injections, you will see some methane. You will probably see some methane increase relative to background concentrations at most sites. What we've typically seen, and this is an example from experiments done by Dr. Borden, is that methane is usually not going to accumulate at levels that would present themselves as an issue for any type of surface regulation or issue. There are, those outlier cases where methane has become an abundant problem, but I would say the majority of the cases out there that I've worked on, I've never seen methane get much higher than, say, a few parts per million in the groundwater. So we're talking molecular levels of methane. I think methane is kind of one of those things that it's sort of a double-edged sword. You want to see some methane because that's an indication that you have fermentation going on. So you do want to see it happen, because if you don't see it at all, it can, and you aren't seeing much TOC, it can be an indication that things really aren't taking off at your site. So you do want to see some, because it's a good metric. What you don't want to see is you don't want to see too much. One thing you can do if you start seeing a lot of methane and you're concerned about your DHC, again, this is something that Frank Loeffler pointed out, is that if you get an overabundance of methane, that means you have a large methanogenesis population. One thing that you can try to do is inject some more B12 at the site, because B12 is going to be one of those cofactors that's important for D. halcocoides and can help sustain its growth in the presence of methanogenesis. So that's something that you could try. Just continuing on with TOC and volatile fatty acids. This is out of the ESTCP AFC protocol that's out there on the website. This is looking at TOC and how TOC and volatile fatty acids track. So as I said, TOC may not necessarily be a good metric long term for performance monitoring of your EVO. So that's where volatile fatty acids come into play. So instead of looking at TOC, and I don't know that I would say instead of, I would say in addition to TOC, you might want to also start measuring volatile fatty acids. Ideally, you'd like to start doing that during your baseline sampling, so you have a background point or a control point to cross-reference your data to, so you kind of see what the changes are relative to that background. But if you are already into a project and you need to come up with something quickly to kind of gauge the performance, you can look at volatile fatty acids relative to TOC. And so that's what this graph does. So they're showing they didn't measure volatile fatty acids at the beginning. They started with TOC, and you can see that 6 months in, they got this huge burst of TOC. That's not uncommon for EVO injections. But then 18 months out, they're seeing it drop way down, almost, getting close to where it was at the beginning. Going from, around 50 or so up to 1,000 is like, wow, that's really good. We got a lot of carbon in the ground. And then, a year later, it's back down to 200. You think that you've burned through all that carbon, and you haven't. See, because when you look at your volatile fatty acids, which is the blue line, you can see that relative to that TOC line, it is several orders higher compared to your TOC. So it's like around 200 for TOC. It's around 600 for volatile fatty acids. So again, if volatile fatty acids is probably going to be a better metric long term for gauging the performance and the life of your injection. Okay, so now I'm going to switch gears a little bit. I just want to touch on bioaugmentation quickly. I gave a whole webinar a couple of years ago all about bioaugmentation and what it can do for your site. This is just going to sort of touch on that briefly because I kind of want to encapsulate all of this in terms of a bio project. The data here, if we look at this graph on the left, this is just looking at a control versus a biostimulated and bioaugmented plot versus just biostimulated. And you can see that when you inoculate the system with the correct microorganisms, you get much more rapid cleanup. They got 92% ethene within a 43-day time period, where at 44 days with just the biostimulated plot, they hadn't generated any ethene. In fact, they were still struggling getting through cis and vinyl chloride. And then if we look at this AFC demonstration project from Kelly Air Force Base, this is actually a field injection with electron donor, and then after they bioaugmented, the reaction that the site had. There's not a whole lot of time in between, about 6 months or so, but it's still sort of an indication that, look at the cyst that they saw. This is the cyst here, this triangle line, and when you compare it to after bioaugmentation, you can see that they got much better results. They dropped that cyst and they started producing ethene. So again, just sort of a kind of validation here that sometimes bioaugmentation is necessary. How do you sort of gauge that? How do you know if bioaugmentation is going to be necessary? Well, that's where your molecular biological tools comes into play. So if you can run a qPCR on your groundwater, this can tell you what organisms are there at what cell concentration. You can even target functional genes of those organisms and tell you, do you have that dihalocoides with the vinyl chloride reductase gene? One limitation that usually isn't said about qPCR is that it's just a count of the cells. It cannot necessarily tell you if those cells are alive or dead. Now, what I understand is that typically, if your cells are really high, you have something to say 10 to the 4th cells per mil, there's a good indication that you have a very viable population. So just because those cells may or may not be dead, typically when you start getting into the very high concentration range, you can make that interpretation that you've got a good, robust population. Something else that's typically done is your phospholipid fatty acid. So now this can verify whether or not that biomass is alive or dead. And the other thing it can do, it can kind of group your organisms into certain communities, say like your fermenters, your sulfate reducers, that kind of stuff. But this can be used along with stable isotope probing to tell you, yes, your contaminant of concern is being used, and a way to sort of definitively prove that bioremediation is occurring, because that biomass will incorporate carbon-13 labeled contaminant into its mass, and that can be tracked with isotope probing. So these are probably the three most common molecular biological tools that are used out there. Most commonly, when people deploy these, they're either using a biotrap, and I won't spend any time talking about those because there's entire webinars about biotraps, but that's sort of like an in situ microcosm that you can deploy at your site. The other way is if you just need to do a quick grab sample. This is typically done for your qPCR, is using one of those bioflow filters, and that's depicted on the right-hand side. I'll just say I got all these images from Microbial Insight, so if you have specific questions, you want more information about what these tests can do for you, I would recommend contacting them. So how do you calculate how much culture you need? It's very similar to the EOS. Basically, we need to know a treatment area and we need to know a pore volume. We also need to know what your target cell concentration is, and for many sites it's the same. It's usually about 10 to the 7 cells per liter that you want your D. halcocoides to be. So what we would do in this theoretical calculation, say you had 200,000 liters of pore volume and you wanted a target cell concentration of 10 to the 7th cells per liter, we would divide that, we would multiply your pore volume by your target cell concentration and then divide that by the cell concentration of our culture, which is 10 to the 11th cells per liter. And then that number would produce a liters of culture. So in this case, it would be 20 liters of the BAC9. A lot of cases, you can just use this ratio. It's like 1 liter of BAC9 per about 10,000 liters of pore volume, if you just kind of want a rough estimate. So everybody is aware, the BAC9 is sold in 19-liter kegs, and those kegs can be concentrated up to 10 times. So if you ordered 30 liters and I send you 19, please don't call me all upset. You got your 30 liters. There should be a hang tag on your keg, and it will tell you, you got 30 liters of culture, the volume is 19 liters, and we concentrated that 1.6 times. So that would equal your 30 liters you got. So in the field, what you need to tell your guys to do is, say you wanted to do 2.72 liters per well. I just made up a number. Well, now what you would need to do is you need to take that unconcentrated injection volume and divide it by your concentration factor, and that would give you your new injection volume. So instead of doing 2.72 liters per well, you would do 1.7 liters per well. And you don't have to write all this down. If you want to order some BAC9, or that's something you want to write into your work plan and you didn't get all that, just shoot me an email. What you need to know about your site is you want your DO levels to be low as you can get them. To be less than 0.5 milligrams per liter is ideal. Obviously, if you just did a massive EOS injection with a bunch of aerobic water, that's going to take some time, maybe a month or so. So that's something you're going to want to watch out for. I usually don't recommend people do in tandem injections with BAC9 and EOS, just because now you got to deal with deoxygenated water, and for some sites that can really be a challenge. For other sites, it's not a big deal. So you just got to weigh it against your site logistics. Sulfate concentrations in the groundwater, they can be a concern. They have a history of being a problem, but we've also got several documented cases where sulfate really isn't an issue, and D. halcocoides is not necessarily competing is not necessarily competing with sulfate reducers, but you can lose some hydrogen to sulfate reducers, so that is something to consider. But again, if your site was properly dosed, it shouldn't be a concern. Also, the CVOC concentrations, again, when I first started doing this, it was kind of a misconception in the industry that really high TCE or PCE concentrations meant that the water was too toxic for bioremediation, and we're finding out that that's really not the case. The biggest issue is going to be controlling your groundwater chemistry in terms of pH. But other than that, D. halcocoides isn't really susceptible to high levels of TCE or PCE. They will plow right on through it. So as long as you can give them the donor, they will have at it. The other nice thing is that the oil does sequester the chlorinated solvents, so you can reduce your groundwater concentrations just by having the oil in there. So that can be something else that can aid in the site's remediation. All right, last topic is pH. I think everybody knows that pH is a problem, especially for dechlorinators. This is very well documented. Here's a graph just showing you that the optimal pH range for D. halcocoides is going to be between 5.5 and about 7.5 to 8. When you get outside of this range, the bug is really not happy. It doesn't grow very well. So your degradation rates are going to start to drop off. So you need to add some type of buffer to the system. Here at EOS, we like magnesium hydroxide a lot just because it gives you a lot of OH equivalents per kilogram. The issue with magnesium hydroxide is that it is not soluble. What's interesting about magnesium hydroxide is its solubility, its dissociation is actually controlled by the pH of the environment it's in. So as the pH gets more acidic, you actually get more dissociation of magnesium hydroxide. It will actually release both of its hydroxide ions, so that way you're just left with magnesium ions. And then as the pH is adjusted, it will actually reprecipitate and can actually reprecipitate completely into its mineral form. So because of this, it has good longevity. How we've sort of overcome the insolubility problem is we've found a proprietary grind of magnesium hydroxide that has a really small particle size, just as small as the oil droplets, about 1 to 2 microns. And then we've made that into a colloidal suspension at 45% using some dispersants and stabilizers, and this helps keep the particles from flocculating together, so you maintain that 1 to 2 micron particle size. We worked extensively with CB&I on, I wouldn't say necessarily developing this product, but sort of on validating this product. So they were some of the first ones that saw the value in using magnesium hydroxide. So the first thing they did was a couple of lab titrations. And really what this graph demonstrates is that 2 different site materials that had similar starting pHs, when they added they got close to about 13 grams of co-buff per kilogram of soil, they achieved their neutral pH, which is this first line here. And that's basically where they needed to stop. But like any good titration, they kept going to kind of see what the upper limit was. And what they ended up seeing is that the titration curve started to flatten out, even though they had added almost double of what they actually needed. So there's this kind of safety net built into the product where you can add a little bit more than you think you might need, and it's not going to be detrimental to the site, unlike some other caustic agents that are used out there like sodium hydroxide. Finally, this is some performance data from that same project. This is from CB&I. They injected this along with some EOS, and what we're looking at here is this graph. This is distance here on the x-axis, distance from the injection point. So 0 would be the injection well. And then we're looking at as a function of pH. And what you can see is at the day 0, each one of these lines is a time interval. We're across the board, we're at about a 4.5, 4.6 pH. But a year later, after they injected the co-buff, this is that blue line. You can see that even out to like 10 feet away from the injection well, they were still maintaining a neutral pH. And you could, even at about 20 feet, they had decent pH readings. It was still around 6. So I think they ended up claiming that they were getting good pH distribution a year later, about 15 feet away from their injection location. So they were very pleased with these results. And we've continued to see these types of results coming in from other people who have used this product. I guess I'll wrap up with this last little bit about pH, and that is, if you want to know how much co-buff you need for your site, you really need to do a soil and groundwater acidity titration. Obviously, you're going to need to couple that with your contaminant concentrations, because the more chlorinated solvent concentrations you have at your site, the more acidity you're going to produce. And then again, going back to your substrate selection, if you're trying to throw a bunch of lactate at this problem and you're concerned about pH, that's something that you're going to need to know right up front, because you'll be fighting that the entire time you're trying to do your remediation. So really, I would say the acidity is probably the most crucial bit. And with that will end my presentation. So I will stay on the line and take any questions that anybody has. You can feel free to type those questions into your question box on your dashboard. I'll be more than happy to answer those. If we run out of time, there's my contact information on the screen. You feel free to email or call me. I do already have a few questions in. And so the first question is do you see dechlorination from bioaugmentation when the electron donor source is low, say 10 milligrams per liter? That's a hard question to answer because it sort of depends on what your electron donor source was. If your electron donor source was EVO, I would say yes, because as I pointed out in the slides at the beginning, you're going to get sorption of that oil. So your TOC in groundwater is going to be low, less than 20 milligrams per liter. And so you could have fine dechlorination rates going on even though your TOC is measured as being low. Now, if your TOC was being measured from a soluble substrate like lactate, I would say probably not. Your site has probably reached a point where you need to think about reinjection. One thing we do know from some work that Dr. Loeffler has done is that I don't believe that there's any really low threshold of electron donor or contaminant concentration in the aquifer. It really just comes down to a contact efficiency problem. I got one question here about, can I comment on calculations for injections into fractured rock environments? Injecting into fractured rock isn't going to be, in terms of the dosing calculations, it isn't going to be too much different from injecting into unconsolidated material. I would say the biggest difference is going to be your oil retention capacity is going to be a lot lower. You're going to want an oil that has a high retention capacity because there isn't all those attachment sites that like there would be in unconsolidated material. That would probably be the only initial consideration that I would have, but that's something that's already built into our design tool. So if this was a site that you were concerned about, we would be able to utilize the characteristics of that fractured rock with one of our emulsified oils and do an appropriate calculation for you. Could you recommend a column study for site-specific oil retention? Actually, if you're interested in measuring your site's oil retention capacity, you can hire, there's a couple of labs that will do that for you. I think Prima will do it, I think Serum will do it, and I think Microbial Insights will do it. The critical part will be collecting a sample that's representative from the treatment interval that you want for injection, because oil retention characteristics can change with depth. They can also change spatially, laterally. So you want to be sure that you're getting a good representative sample of your site material. So that's something that you can hire a lab to do. Otherwise, if you want to do it yourself, you could order some product from us. You could dilute it and inject it through a column of material that you've constructed yourself. After you've passed that material through the column, you flush it with some clean water, a couple pour volumes, and then you can send sections of that material off for, say, like a loss on ignition or something like that, some way to estimate how much carbon was burned off. Have we used the product for remediation of saturated soils? Yes. The nice thing about EOS is that as long as you actually have some saturation in your soils, the product's going to work. Typically where we run into an issue is when we're dealing with unsaturated soils. But we do have a product called VOS that's for the Vados zone. And basically what that does is it sets up a sort of a perched in situ microcosm cell of emulsified oil and dehalcocoides, so that way you actually can do enhanced bio in the unsaturated zone without worrying about the product leaving that zone. It sort of sets up like a gel. I think that's all the questions that I have right now. If anybody has any more questions, I'd be more than happy to answer those. I don't see any more questions coming in, so what I'll do is I'll just go ahead and end the webinar for today. We're at the 1 o'clock hour anyway, so I really appreciate everyone attending today. Thank you so much. And if you do think of more questions, or there was maybe something you weren't quite sure about right now, but it comes to you later, feel free to give me a call or shoot me an email. Be more than happy to help anybody out with any issues they may have. So with that, thank you very much. Have a great afternoon.